في محاضرة اليوم هي عبارة عن introduction to endocrinology. راح ناخذ بعض الأمور المهمة اللي راح نعتمد عليها فيما بعد. بسم الله. Endocrinology. When we say endocrinology, what do we mean by that? Endocrinology is the study. In the endocrinology, we study the hormone, its receptor, and its signaling pathway, as we will see. So endocrinology is the study of the hormone and its receptor and its signaling pathway. As we will see, when the hormone binds to its receptor, this binding will create some signaling pathways. So the hormone and the receptor and the signaling pathway all together, we call them endocrinology. And as you studied before, the glands in general are divided into two types. Glands with ducts, as of the GIT, the one you studied, and the glands without duct, ductless. We call them endocrine glands. So endocrine glands are ductless glands. Their secretion, we call it hormones. Hormones are chemical messenger. It's communication between the cells. So the cells communicate through hormones. And as we will see, hormones has a wide variety of function. In the reduction, in regulation of internal environment, in the growth, metabolism, and so on. In our body, this is the endocrine glands found in our body. The pituitary gland, thyroid gland, adrenal gland, testis, ovary, pancreas, and so on. First, we will classify the hormones according to the distance they travel from the place of secretion until they bind to their receptors on the target cells. Endocrine, according to the distance, or the hormones, according to the distance they travel, they are divided into three groups: endocrine, paracrine, and autocrine. In the endocrine, the hormone released by the gland, the endocrine gland, the hormone enter to the main bloodstream that transport the hormone for a distance away from the endocrine gland that released it. So in the endocrine, the hormone released and enter to the main bloodstream that travel a distance away from the place of its secretion. While in the paracrine, the hormone does not enter the main bloodstream. No, it enters to the local circulation within the tissue only, to the local circulation. This is the paracrine. While in the autocrine, the hormone affects the same cell that releases it. See the difference? So in the endocrine, the hormone enters to the main bloodstream. Thus, it travels a distance from the place of its secretion. While in paracrine, the hormone enters the local circulation. It does not enter the main bloodstream. And in the autocrine, the hormone affects the same cell that released it. This is the three groups according to the distance that the hormone travels. But what is important in our biochemistry is the classification of hormones chemically. Chemically, hormones are classified into three groups. Chemically, hormones are classified into three groups. The first is steroid hormones, second amine hormones, the third is peptide and protein hormones. So chemically, this is the classification of hormones. 3 groups. The first group is the steroid hormones. As we said before in the GIT module, if you remember, we said all steroid, this is a must you should know. All steroid hormones are synthesized from cholesterol. All steroid hormones contain 21 and less of carbon atoms. This is the steroid hormones. So all steroid hormones are synthesized from cholesterol. All steroid hormones contain 21 or less of carbon atoms. Thus, steroid hormones are classified into 6 classes. Progestin, they contain 21 carbon atom. Example, progesterone. Glucocorticoids, 21 carbon atoms. Example, cortisol. Mineralocorticoids, 21 carbon atoms. Example, aldosterone. Androgens, 19 carbon atoms. Example, testosterone. Estrogen, 18 carbon atoms. Example, estradiol. And finally, vitamin D. Vitamin D because it is synthesized from cholesterol. Thus, steroid hormones play a very major roles in carbohydrate metabolism, in salt, water, and reproductive function. Because 6 hormones, the 6 hormones are steroid hormones. Second group of hormones are amine hormones. You should know about amine hormones that all amine hormones are synthesized from the amino acid tyrosine. So amine hormones are the hormones that are synthesized from which amino acid? Tyrosine. When we say amine hormones, it contains 2 classes of hormones. This is catecholamine hormones, the first, and thyroid hormones are the second. So amine hormones include which hormones? Catecholamine hormones and thyroid hormones. When we say catecholamine, we mean epinephrine, norepinephrine, and dopamine. Epinephrine, which is the adrenaline, the other name for adrenaline, and noradrenaline and dopamine. So we can, adrenaline has, you can call it adrenaline or epinephrine. Same thing to norepinephrine and dopamine. So catecholamine include 3 major hormones: epinephrine, norepinephrine, and dopamine. While the other classes that is synthesized, that is in this group, amine hormones, is thyroid hormones. As we will see in our 5th lecture, thyroid hormones are synthesized from tyrosine amino acid. And catecholamine hormones include 1 tyrosine ring, as we will see. While thyroid hormones are synthesized from 2 fused rings of tyrosine. So to synthesize catecholamine, there is a need for 1 tyrosine amino acid. While the thyroid hormones, there is a need of 2 tyrosine rings to synthesize the T3 and T4 thyroid hormones, as we will see in our 5th lecture. Now we will study a little bit about amine hormones, the catecholamine. As we said, amine hormones include catecholamine and thyroid hormones. Thyroid hormones, we will study it in a little bit of details in the 5th lecture. Catecholamine, we will take a very fast synthesis pathway for catecholamine. This is the catecholamine hormones. This is the catecholamine hormones. As we said, catecholamine hormones, as you can see in the structure here, starts the synthesis from which amino acid? Tyrosine. This is the tyrosine. To your left hand, always the name of the process, hydroxylation of the tyrosine ring. To your left hand is the name of the enzyme that do this process. So The synthesis of catecholamine hormones starts with single tyrosine amino acid. The first process is the hydroxylation of tyrosine ring. Hydroxylation, the name of the process. So what will happen? An addition of one hydroxyl group to the ring of tyrosine. What is the name of the enzyme? Tyrosine hydroxylase. So tyrosine hydroxylase add a hydroxyl group to the ring of the tyrosine. Thus, tyrosine became dihydroxyphenylalanine, shortly known as DOPA. So DOPA is the short form for dihydroxyphenylalanine. Then DOPA is converted to dopamine through a process known as decarboxylation. So decarboxylation, what will happen? Removal of decarboxylation, removal of carboxylic acid group, COO. What is the name of the enzyme that do this process? Aromatic amino acid decarboxylase. So aromatic amino acid decarboxylase break the carboxylic acid group, thus DOPA is converted to dopamine. This happens in the cytosol. Then dopamine enter inside. So the first catecholamine to be synthesized is the dopamine. Then dopamine enter inside the storage vessel. Then the next process is the hydroxylation of the side chain of dopamine to convert the dopamine into norepinephrine. So dopamine is converted to norepinephrine through what is the name of the process? The hydroxylation of the side chain. What is the name of the enzyme? Dopamine beta hydroxylase. Then norepinephrine is converted to epinephrine through methylation. So when methyl group is added to norepinephrine, then norepinephrine is converted to epinephrine. What is the name of the enzyme? Phenylethanoamine and methyl transferase. So this enzyme add a methyl group to norepinephrine, thus it converted to epinephrine. This is a very short synthesis pathway for catecholamine hormones. So as you can see, all catecholamine hormones has nearly similar synthesis pathway, and this is they all started from a single tyrosine amino acid. The third group of the hormones, as we said, is the peptide and protein hormones. Most of the hormones in our body belongs to peptide and protein hormones. So most of the hormones belongs to peptide and protein hormones. As you know, to synthesize a protein or a peptide or a polypeptide, to synthesize, there is a need for activation of a gene. There is a need for transcription and translation. And this translation occurs on the ribosomes, as you can see here. And ribosomes, they may be found in a single ribosomes separately in the cytoplasm, or normally they are found in the endoplasmic reticulum on the surface, as you can see. This is why we call the endoplasmic reticulum that has ribosome on its outer membrane stuck to it, granule endoplasmic reticulum, or sometimes we call it also rough endoplasmic reticulum. So rough or granule endoplasmic reticulum are endoplasmic reticulum that has ribosomes on its surface. So there will be transcription. The transcription will make messenger RNA. Messenger RNA will leave the nucleus and will come and stick to the ribosomes in the granule endoplasmic reticulum, where the translation occurs. The translation will build the first hormone, which is known as pre-prohormone. The pre is a number of amino acids. Prohormone, so the hormone is not a mature hormone here. The first, it is synthesized, the protein and peptide hormones. Once they are synthesized as pre-prohormone. Pre is a number of amino acids. Once the hormone, the pre-prohormone enter inside the endoplasmic reticulum, a number of amino acids that form the pre-section are cleaved. Thus, the prohormones enter inside the endoplasmic reticulum. Then it is transported to Golgi's apparatus, and the prohormone is stored in the secretory vessels and remain there as a prohormone until it is stimulated for the secretion, as we will see. Then once before the secretion, it is activated to hormone. And its activation is very similar to the enzymes, the zymogenes, if you remember what the one we studied in the GIT. So zymogenes are activated only once needed. Prohormones are in the secretory vessels. Before they are released, they are activated into hormones, which are released into the bloodstream. This is the synthesis for peptide and protein hormones. As you can see here, this is the protein and peptide hormones. They are peptide. They are made up of amino acids. There is a need for their synthesis. There is a need for transcription and translation. And they are first synthesized as pre-prohormones. Steroid hormones, all steroid hormones are synthesized from cholesterol. Tyrosine, the amine hormones, as you can see, catecholamine hormones such as epinephrine are synthesized from single tyrosine ring. While as you can see, thyroid hormones as of thyroxine, the T4, is made up of two fused rings of tyrosine. The one we will study in our fifth lecture, inshallah. Now, control of hormone secretion. What control the hormone secretion? What stimulate the secretion of hormones? First, neurons. Example, the adrenal medulla, which is the inner part of the adrenal gland, which secretes the epinephrine and norepinephrine in fight or flight situation, as we will see. So, neuron control the secretion of some endocrine glands, such as the adrenal medulla. While also the second stimuli is hormonal. So some hormones, as we will see also, stimulate the secretion of other hormones. And lastly, humoral. Humoral means substances. For the best example is the high blood glucose stimulate the secretion of insulin. So glucose in our blood, the high level, stimulate the secretion of insulin. So what stimulate the secretion of hormones are neural, hormonal, or humoral. Feedback control. Feedback control is very similar to the one you studied in your first year in the enzyme. In the enzymes. The feedback mechanism that when the products accumulate, they will stop the first step of the reaction. This is the negative also. Negative feedback mechanism. When there is no accumulation of the products, as we will see in this example, the secretion will continue. This is a positive control. So we will take of one example about the feedback mechanism. Means, what do we mean by feedback? Now the hormone is secreted, or the endocrine gland secreted or started to secrete the hormone. What will stop the secretion now? We said in the last slide, we said that there is 3 factors or 3 things that stimulate the secretion of hormones. Now, what stops the secretion of hormones? The feedback mechanism. The feedback mechanism. For example, we will study the thyroid hormones feedback mechanism. First, the hypothalamus. As you will see, once you progress, it controls most of the endocrine glands. Most of the endocrine glands is controlled by the hypothalamus. Always, we call the secretion of the hypothalamus the releasing hormone. Releasing hormone. Because we are talking about the thyroid hormones, it is called thyrotropin releasing hormone, shortly known as TRH. So the hypothalamus releases thyrotropin releasing hormone, the TRH, that stimulates the pituitary gland to release thyrotropin stimulating hormone, always pituitary gland stimulating hormone. See, as we said, what stimulates the release of hormones? As we said, the neurons and hormones. As you can see, what stimulated the TSH? The TRH. So hormones stimulate the release of other hormones. Also, the TSH, thyrotropin stimulating hormone, stimulate the thyroid gland to release T3 and T4. The one we will study in more details in our 5th lecture, inshallah. Now, so the hypothalamus releases TRH that stimulates the pituitary gland to release TSH. TSH stimulates thyroid gland to release T4 and T3. This release will continue. It will continue until the level of T3 and T4 in our body is high. Then comes the negative feedback. What is the negative feedback? When the T3 and T4 level is high, when their level is high, they block the release of TSH and the TRH. This is the negative feedback. So high levels of T3 and T4, when they accumulate in high level in our blood, they will stop. They will block the TSH from pituitary gland, the TRH from the hypothalamus. Thus, the synthesis for T3 and T4 will be ceased, will stop. This is the feedback mechanism for controlling the release and synthesis of hormones. And this is how some hormones are controlled. Now, our next is hormone synthesis, storage, secretion, and transport. First, we will talk about protein and peptide hormones. As we said, for the synthesis of protein and peptide hormones, there is a need for stimulation of some genes, and we will study in our 4th and 5th lecture how the hormones also stimulate some genes, how they control the activity of some genes. So, for the synthesis of protein and peptide hormones, there is a need for transcription, then there is a need for translation of these messenger RNA on the rough endoplasmic reticulum. And as we said, the synthesized pre-Bru hormones, which is then cleaved to be Bru hormones, is stored inside the secretory vessels until needed. Once there is a stimulation for this endocrine gland to release its hormones, there will be the secretion. When there is a stimulation for this, whether it is a neuron or humoral or hormones, once there is a stimulation for this endocrine gland to release its hormone, the permeability of calcium ions increases. Thus, this calcium ions enter inside these endocrine glands and cells. It causes the activation of some enzymes and microfilaments and microtubules, which causes the fusion of the secretory vessels with the cell membrane and the exocytosis. Let us go back just to show you this here. So the prohormone now is stored in the secretory vessels. Once there is a stimulation, what will happen? Calcium ions permeability will increase. The calcium ion will enter inside inside these cells in the endocrine gland, which will cause the stimulation of some enzymes, microfilaments and microtubules, which will move. See, it will move these secretory vessels until it reaches the cell membrane and fuse with it, and thus the release, as you can see, the release of the hormones to the bloodstream. This process, we call it exocytosis. Exocytosis, when the cell releases things to the outside, we call it exocytosis. When it enters inside from outside to inside, we call it endocytosis. Endocytosis, while releasing is exocytosis. So, as you can see, peptide and protein hormones are stored in the secretory vessels. Then, when these cells are stimulated, calcium ions enter inside these cells in a large amount, causes the stimulation of some enzymes, microfilaments, microtubules, which these together will move the secretory vessels until it is fused with the cell membrane, which will cause the release of the hormones inside them to the bloodstream. This, we call it exocytosis. We call it exocytosis. This happens to the peptide and protein hormones. Second is the steroid hormones. Steroid hormones. In the steroid hormones, they are synthesized in the mitochondria and endoplasmic reticulum. See, what is the difference? They are synthesized inside the mitochondria and endoplasmic reticulum. Because if you remember, we said there is a need for B450 side chain cleavage to cleave the 27 carbon atom cholesterol to become prognolone, the 21 carbon atom. All steroid hormones are synthesized from cholesterol, and when cholesterol loses 6 carbon atoms, we call that structure prognolone. Then all steroid hormones are synthesized from this prognolone, the 21 carbon atoms. And also inside the mitochondria, there is 2 electron transform enzymes, ferredoxin reductase and ferredoxin, that they will help in the synthesis of steroid hormones. But see the difference in the storage. There is no storage for steroid hormones. So the endocrine glands that release steroid hormones does not store the steroid hormones. Once the steroid hormones is synthesized, it is released directly into the bloodstream. This is the difference. So peptide and protein hormones are synthesized by an order from the gene through transcription and translation, and they are stored, and then there is a secretion. But in the steroid hormones, they are synthesized in the mitochondria and endoplasmic reticulum, and they are not stored. Once they are synthesized, they are released directly to the bloodstream. Transport of the hormones. One important or very important thing you should know from today's lecture. Because inshallah in tomorrow's lecture we will start with the plasma membrane receptors. Hormones also are divided into two groups according to their hydrophobicity. Some hormones are hydrophobic, some hormones are hydrophilic. Steroid hormones and thyroid hormones are hydrophobic. So, steroid hormones and thyroid hormones are hydrophobic, while catecholamine hormones and peptide and protein hormones are hydrophilic. This information is very important. You should know. So, steroid and thyroid hormones are hydrophobic. This is why, as we said in the GIT, in the absorption of the food from the small intestine, hydrophobic, they need a transporter in our blood. This transporter, what is the transporter for the hydrophobic hormones? Are the plasma globulin and albumin. And sometimes there is also specific transporter, such as steroid hormones binding globulin, shortly known as SHBG, and thyroid hormones binding globulin, TBG. So, the hydrophobic hormones, steroid hormones, and thyroid hormones, they need a transporter. They either is transported directly with the plasma globulin or albumin, or some specific transporter such as steroid hormones binding globulin SHBG or hormone binding globulin TBG. See here. So as you can see in the steroid hormones and thyroid hormones, they are bonded with a transporter, globulin or albumin or SHBG or TBG. As long, this is important also, as long as the hormone is bonded with its transporter, it is not active. What is the active hormone? Is the free hormone. So what is the active hormone? Is the free hormone. As long as the hormone is bonded with its transporter, it is not active. It is not active. This is why there is a balance between the bonded hormone and the free hormones. The free hormones are the active one, while the bonded hormones, they function as a storage for free hormones. When the concentration of the free hormones become low, then free hormones are released from their transporter to become a free hormones. So there is equilibrium, there is a balance. We have, for example, we have thyroid hormone binding globulin, thyroid hormone binding globulin TBG. This transporter binds thyroid. Is this thyroid hormone active? No. Why? Because it is bonded with its transporter. On the other side, there is a free thyroid hormones. When this thyroid hormone binding globulin TBG, when it releases the thyroid hormone, it releases its thyroid hormones once the free thyroid hormones level became low. So the bonded hormones function as a storage for the free hormones. This is very important concept. Neuroendocrine interaction means there is an interaction between neurons and endocrine. There is interaction. As you can see here, see, this is the best example about a neuron and the endocrine interaction is between the hypothalamus and the pituitary gland. See, who controls the pituitary gland? The hypothalamus. Hypothalamus is a neuron. Is neurons. It's made up of neurons. It controls what? The pituitary gland, which is endocrine gland. This is the interaction between neuron and the endocrine gland, and this is the best example about their interaction. Now, the concentration of the hormones is controlled by 3 factors. The endocrine glands is stimulated to release hormones. Then the release of these hormones from the endocrine glands is controlled by feedback mechanism. Then the feedback mechanism will stop the release of the hormones from the endocrine glands because there is enough levels of the hormones in the blood. Now, what happens to the hormones in the blood after they do, for example, after they do their function? What will happen? As we said, what determines the concentration of the hormones in our body, as we said, is the rate of the production, the feedback mechanism. It does not allow continuous level or secretion of the hormones. No, there is a feedback mechanism. Second, what controls the concentration of hormones in our blood? Rate of delivery. Rate of delivery to the specific organ, to the target cells, which relies on the rate of blood flow to that organ. So, how fast the hormone reaches the target cell relies on what is the rate of the flow of the blood to that organ. If the blood flow to that organ is high, then the delivery of that hormone to that target cells will be high. The third that determines the concentration of hormones in our cells is the rate of degradation. Rate of degradation. Normally, the main organs that break down the hormones is liver and kidney. Also, the target cells also degrade some of the hormones. So the concentration of the hormones are determined by rate of production, which is controlled by the feedback mechanism. Rate of delivery, which is controlled by the blood flow to the target cells or target organ. And the rate of degradation and elimination, which is controlled or cleared by the liver, kidney, and the target cells. The last thing in today's lecture is the last topic is spare receptors. See here. What do we mean by spare receptors? For example, one cell has 100 receptor for thyroid hormone. How many receptors? 100 receptors. Now, is there a need to 100 hormone to stimulate the 100 receptor? The answer is no. Normally, there is a need to stimulate. It's difference between one hormone and another. This is an example. There is a need normally to stimulate 2 or 3 hormones, 2 to 3% of the receptors to get full response from the target cell. So although the cell has 100 receptors for the thyroid hormones, but stimulating 2 or 3 of these receptors is enough. So 2 or 3 receptors are enough. So now the 97 receptors, we call them spare receptors. So the cell means has excess of number of receptors that we don't know its function actually. We'll talk about it in a few minutes. So normally stimulation of 2 to 3% of the receptors are enough to reach 100% response from the cells. What happened if the 100 receptors are stimulated? Same response. So if you stimulated 2 to 3%, or you stimulated the 100% receptors, same response. So the 2 or 3% are the required one. They must be stimulated. While the 97 or 98%, the remaining, we call them spare receptors. What is the function? It is hypothesized. Means scientists think that spare receptors mean that the cell has much more receptors than it needs to increase the chance of meeting between the hormone and its receptor. Because just imagine that the hormone is distributed by the blood to all our body cells. To increase the chance of meeting between the hormone and its receptor, the cell normally has much more receptors than it needs. This is a hypothesis for scientists. Second thing, scientists also say that suppose that there is a very limited number of hormones, very few. So the hormones will jump from one receptor in the cell to another. So it stimulates one receptor, then it will jump to the other receptors on the same cell, then to the third, and so on, until the cell gets the 100% response. This is the hypothesis why there is spare receptors, but the main idea is to increase the chance of meeting between the receptor and its hormone. This is in this graph, as you can see, for this enzyme or for this example, there is a need, there is a need to stimulate 50% of the receptor, as you can see. When there is a stimulation of 50% of the receptors, there is 100% response. What happened if there is a stimulation of 100% of the receptor? Same response. So whether you stimulated 50% or 100%, the same response. So the required are 50% and the spare are the other 50. This is just for you, I think 100%, not think, 100% you study it, just some vocabulary. Agonist is the molecule that binds to the receptor and induces full response or stimulation. While antagonist in the shape is very similar to agonist, but it does not stimulate the cells or for any effect. This is our today's lecture. إذا عندكم أي سؤال تفضلوا.
دكتور أنا عندي سؤال.
تفضلي.
دكتور هلا الهيدروفوبيك هرمون
تفضلي.
عشان ينتقلوا بالدم بدهم ترانسبورتر صح؟
نعم.
طيب عشان يصير لهم اكتيفيشن لازم يصيروا فري.
نعم.
طب كيف يعني بدهم يكملوا مشي بالدم وهم اصلا هيدروفوبيك؟
لا بكونوا يعني لازم تلاحظوا انه يعني بكونوا قريبين جدا من التارجت سيلز، يعني ما بكونوا بالاندوكراين. الان خلينا نحكي steroid hormones are released. Steroid hormones are released now. بيرتبطوا مع الناقل تاعهم، الناقل تاعهم بينتقل للتارجت سيلز very close to the target cells. This is why there is a balance. مشان هيك حكينا balance. يعني بيكون هناك sense between the three. هذا كيف الرابط وشو اللي بده يعمل له stimulation، يعني العملية معقدة جدا. كيف الرابط أو الناقل هذا بده يعرف إنه التركيز الفري هرمون صار قليل جدا، فهناك سنس وهناك انزيمات تتحكم بهذه العملية، وهناك ضبط كبير جدا في جسم الإنسان لهذه الأمور اللي هو يكون هناك balance between the free hormone and the bonded hormones. And the free hormones and bonded hormones are very close to each other. Very close to each other. نعم. عندكم سؤال؟ في أسئلة؟ شكرا لكم لحسن استماعكم، بارك الله بكم، وإن شاء الله بنلتقي غدا بالمحاضرة الثانية. المحاضرة الثانية والثالثة نازلة على الريكورد، نازلة قصدي على الموقع عندكم بالاندوكراين. أرجو إنه تدرسوها، أرجو إنه المحاضرة وخاصة المحاضرة 2 و3 أرجو إنكم تدرسوهم اليوم مشان بكرة يكون عندكم فكرة عنهم. لازم تدرسوهم اليوم مشان تسهل عملية فهمهم. السلام عليكم.